Bio 225. It is May 25th, 2019. I am doing the lab for microscope use and function for microbiology. This is my lab report to give me ideas on what I did for the lab. The first part of the lab was going over the different parts of the microscope. So I have the eyepiece and the main tube and the objective lenses, and they rotate around. There's a scanning lens and the low power and the high power and the oil immersion. You move the slides up and down with longitudinal and transverse control knobs. This is the base. This is the illuminator that you turn on so you could see the slides. The other things that we went over were the magnification. This is the ocular magnification, and it's 15 times. The scanning power is 4 times. The low power is 10 times objective magnification. The high power is 40 times. And the oil immersion lens is 100 times. And you multiply those by the ocular magnification, which is like for, let's say, for the oil immersion, ocular magnification is 15 times. So oil immersion would be 100. So 15 times 100 equals 1500, and that would be the total magnification. I'm switching the scanning lens to the oil immersion lens to give you an example of what I had to do for looking at slides. The other thing we needed to learn was field of view measurement. So I put a measuring ruler underneath the scanning power, and it measured the millimeters. So scanning power was 2 millimeters, and the field of view in micrometers, which is what we measure in, is 2,000 micrometers for that. And for our oil immersion lens, it was 0.1 millimeters that I measured under there. So equal to 100 micrometers was the field of view. The next part was scanning different types of slides. So one of the things that I found was hard for the oil immersion lens. It's very close to the specimen when you turn to it. So I found if I lowered or raised up the main tube with the coarse adjustment knob and turned the oil immersion after I put the oil drops on the slide and then lowered it to focus, it wouldn't scrape the slide, because initially I had gotten all my different lenses and focused on it and then changed to the oil immersion, and it was too close to the slide when I turned it, and it scraped the slide a little bit. So it damaged a little bit of the E-slide. That's one of the things that I found that was helpful during the lab to visualize things. The other thing I found was when we did our preparing the wet mount slide, which what we did was we took a slide, we took a Q-tip of a sterile Q-tip, wiped the cheek, and then I dotted it on here. Then I took a drop, but I needed to make sure that I used a very small drop. And then a cover slip, and when I put the cover slip on, I just let it go, and then I used a paper towel to dry the edges. But I needed to be careful not to move the cover slip around, and if you use too much water, the cover slip slides too much off. So it's really important just to use a very little drop on there, and then you just put it on there. I found also not having the illuminator light too bright underneath helped me visualize the slide much better, because it wasn't stained, so it was hard to see my cheek cell smear and the dental tartar smear. So those were interesting to see. The best way I could see them was under the oil immersion lens, this lens right here. So that the only difference, or they were both slightly different but had similar cells, but the dental tartar smear had like little square type things that I could visualize on there, and I'll show you on the slide.
These are the two wet mount slides that I created. This first one is a dental smear. Notice the little squares. This next one is the cheek smear with no squares. Thank you for watching my video.